Okay, good afternoon, everybody. Today we'll discuss the week 3 portion. Just a minute, I'll share my screen first. Hmm. Okay, so hmm. so today we are going to discuss the topics that were covered in the week 3 lecture, and in week 3 it was about RNA transcription. So in this particular lecture is very important because here it talks about how the transcription takes place and how the various post-transcriptional modifications takes place, what are the different enzymes which are involved in RNA transcription. And this is very important not only for your this particular lecture point of view for this coursework, but also for your competitive examinations per se. Okay, so in this week we had a lot many lectures. Lecture 1, 2, 3, 4, 5, I think 5 lectures were there. So first, starting with the central dogma. So what is the central dogma? So central dogma means DNA gets converted to RNA, and RNA gets ultimately converted to protein. This is the central dogma of life. Okay, so when we talk about the central dogma, it is DNA to RNA to protein. DNA to DNA is known as replication. DNA to RNA is known as transcription, and RNA to protein is known as translation. Okay, is it clear? Now, if we see the enzymes which are involved in this central dogma, so essentially there are four important enzymes which are involved in making this central dogma of life. Okay, so the first enzyme is DNA dependent DNA polymerase. So what is the function of this DNA dependent DNA polymerase? So DNA polymerase, that means it is polymerizing DNA and it is dependent on the DNA template. That means it is required for the replication process. So if you see here, this DNA, if this DNA has to replicate itself, it requires an enzyme which is DNA dependent DNA polymerase. So this is the first enzyme which is required for the replication of DNA. Then what is the second enzyme? Second enzyme is DNA dependent RNA polymerase. So RNA polymerase means it is polymerizing RNA. This is clear. DNA dependent means that it is using DNA as the template, and by using DNA as a template, it is synthesizing RNA. So this is required for transcription. Okay. This is required for replication. This is required for transcription. Then RNA dependent RNA polymerase. So RNA dependent RNA-C, wherever it is written this dependent, this means this is required as a template. This DNA polymerase require DNA as the template. This RNA polymerase require DNA template. This RNA polymerase require RNA template, because every enzyme needs some kind of a template, right? So here this RNA polymerase requires RNA template. That means if RNA has to replicate by itself, here you can see if RNA needs to replicate, it needs RNA dependent RNA polymerase. So here your template would be RNA. RNA dependent RNA polymerase. Replicate, it means this is required for the replication of RNA. Then next is reverse transcriptase. Now reverse transcriptase, see, RNA dependent DNA polymerase. What does it mean? It is a DNA polymerase. That means it is required um, uh, uh, it is polymerizing DNA because it's a DNA polymerase. But it is RNA dependent, which means that it requires RNA as the template. Okay. So RNA dependent DNA polymerase means that it is synthesizing DNA but using RNA as a template. So what does it mean? Synthesis of DNA can be can occur with the help of DNA as a template, that is known as replication. But here, if we are using RNA as a template and then when we are synthesizing DNA, this means it is just the opposite of transcription. Okay, so this is reverse transcription. This is reverse. And this enzyme, in short, is also known as reverse transcriptase. Okay, so these are the important enzymes which are required for maintaining the central dogma of life. Now, some important points to understand when we are studying about this central dogma is that the two strands of DNA are not identical to each other, but they are complementary to each other. So, if what does it mean? Suppose if this is your DNA, it is A, T, C, G, T, A. Suppose if this is your one strand, your next strand will not have the exact same sequence, but it will have the complementary sequence. That means your next strand will have T, T, A, G, C, A, T. So what is this? It is complementarity. This is complementarity. So this is the complementarity which is happening. So that is what they are saying, that the two strands of DNA are not identical, but they are complementary to each other. And retroviruses have RNA genome, okay, stop codons. So these are the three stop codons: UAA, UAG, and UGA. These are the stop codons. So whenever stop codon comes, your synthesis of protein stops. And what is a sense codon? Where amino acid comes in, it is known as a sense codon. So there are, just a minute, two types of codons. Hmm. There are two types of codons. One is a stop codon and other is a sense codon. So sense codon is the one which lets the incorporation of an amino acids, whereas stop codon is the one which stops the translation process. Fine. Then now we should know the difference between genotype and phenotype. Before we proceed further, there are two different terms and we should know the difference between them. The first term is genotype and the second one is phenotype. So genotype, what is genotype? Genotype is the genetic content of an individual. The complete genetic information of an individual is known as genotype. Okay. And what is a phenotype? Phenotype is the expression of the genotype that is observed, that we can observe through our eyes. That is your phenotype. So genotype is the genetic content and phenotype is the expression of the genotype. Okay. So genotype expresses and gives us the phenotype. So this is the difference between genotype and phenotype. Okay. Now, when we are talking about this transcription essentially we talk about the formation of mRNAs. Okay, so we are saying that DNA gets converted to RNA. That is your transcription. That is the formation of RNA through by taking DNA as a template is known as transcription. Now there are different kinds of RNA. This is not just that there is only one kind, one RNA. There are different types of RNA molecules. So when we talk about transcription, majorly or by default we are talking about the mRNA transcription, but this central dogma not just not covers the mRNA transcription, it also covers your tRNA and ribosomal RNA transcription also. But essentially, when we talk about transcription, we by default talk about mRNA transcription. Why? Because that only leads to the formation of proteins, and we are majorly concerned about proteins. But the formation of mRNA or tRNA or rRNA from DNA template, all of these three are known as transcription only. And when your DNA forms mRNA or tRNA or rRNA, it is a central dogma. This is a direct central dogma, direct formation of those RNA molecules which are important. There is some indirect central dogma also, where other kinds of RNA molecules which are uh, not important for the protein uh, coding, but they are important for, they have some other roles, that they control the rate of transcription or they control the rate of protein formation. These are small nuclear RNAs, microRNAs, and piRNAs. These are some other RNA molecules. So if we talk about mRNA, so mRNA is the intermediate that carry genetic information from DNA to the ribosome. So this is DNA. It has all the sequence A, T, G, C, G, T, whatever sequence is there, it is there in the DNA. This sequence has to be taken up by the ribosomes. Okay, because when this sequence is taken up by the ribosomes, then only ribosomes can form the protein. Now DNA cannot directly take the sequence to ribosome, so it requires an intermediate, and that intermediate is your mRNA, and which is messenger RNA. So this acts as a messenger as it carries the message from the DNA to the ribosome. That is why it is known as messenger RNA. This is one. Then there is tRNA, adapters between amino acids and codons in mRNA. Now, this mRNA has the information. All the information it has. It has the codon. The codon sequence it has. So this codon sequence has to be read by the ribosomes. Suppose if there is ATG, ATC is there, CCA or whatever is there, this sequence has to be taken up by the ribosome. So this taking of the sequence to the ribosome is, or transferring the information to the ribosome is known as transfer RNA. So this is your tRNA. Then rRNA, it is the ribosomal RNA. What is the meaning of ribosomal RNA? That means that it is present in the ribosome. This rRNA is present in the ribosome and it is required for the structural and catalytic function which is required for the synthesis of a protein. So when a protein is synthesized, then during the formation of protein, ribosomal RNA helps, which is present in the ribosomes. And then these are some other RNA molecules like snRNA, microRNA, and piRNA. So these are all non-coding RNAs. This is also non-coding. Coding RNA only means that RNA which codes for protein. All other RNA which does not code for protein are non-coding RNAs. So earlier it was assumed that non-coding RNA are the junk portion or the intronic sequences which has the non-coding RNA. Those are the junk sequences, but now later, eventually, it was observed that this is not junk sequence. It indeed also has some functional role during the synthesis of mRNA or proteins because it regulates the amount of transcription that happens. Okay, it controls the efficiency of transcription. So now we know that that is not a junk portion, that it also has some role. Okay. Now, so this was all what which was covered in lecture 1. Then lecture 2 was RNA transcription, how RNA transcription takes place. Just a minute. So, uh, just a minute. What happened? Okay, I think we got some problem here. Hmm. Okay, there was some problem. Hmm. So now we're talking about RNA transcription. So RNA transcription, what is the transcription? What is the basic definition of transcription? Transcription involves the synthesis of single strand RNA transcript, which is complementary to one strand of DNA of a gene. That means that this is your DNA. Suppose if this is your DNA molecule, it is double stranded. Okay. This is a double-stranded DNA. In this double-stranded DNA, only one of the strands takes part in transcription, which means that unlike in replication, where both the strands individually takes part in replication process, in transcription, only one of the strand of the DNA takes part in the transcription process. That is what is it is written that transcription involves the synthesis of a single strand RNA transcript complementary to one strand of a DNA. And then what is translation? Translation is the conversion of information which is stored in the nucleotides in the RNA transcript into the sequence of amino acids according to the specifications which are written in the genetic code. Okay, so if we see gene expression, so RNA synthesis, when we talk about RNA synthesis, so you see single and just hmm, see here, this is your DNA. So one is your template, other is your non-template. Okay, so only one of the strand takes part in RNA synthesis. Single strand of RNA takes part in RNA synthesis, and then RNA is formed. And when transcription happens, there is a transcription bubble that is formed, which is very much important for us to understand. Why this is important? Because this transcription bubble is actually the place where the transcription happens. Okay. So transcription bubble is a molecular structure which is formed during DNA transcription when a limited portion of the DNA double helix is unbound. So just like I told you that only one of the strand of DNA takes part in transcription. So does it mean that the two DNA strands have to be denatured and then the process takes place? No. So unlike in replication, what happens that before replication process actually starts, the two strands of the DNA, they are unwound, they are denatured. Okay, and when they are denatured, they completely get separated as two single strands, and then those two single strands individually takes part in the replication process. Therefore we get 2 to the power n. From 2 we get 4. How we are getting 4? Because each strand is synthesizing one. So like this it happens. But here, in the case of transcription, the double helix is not completely denatured. Instead, it gets sequentially unwind. And as it gets unwind, one of the strand gets involved in the transcription process. When that particular portion of DNA is transcribed, then this bubble moves further. So this bubble keeps on moving like this. Okay, in like this bubble keeps on moving. So as and when the transcription is required, this bubbles is formed, and then this bubble starts moving along the direction of transcription. So this bubble where you can see that you have your unwounded DNA, you have your mRNA which is also formed, and your RNA polymerase is also there. This whole thing is known as transcription bubble. So So in prokaryotes, RNA polymerase binds specific nucleotide sequence plus transcription factors and then DNA unbinding. Now, very important thing is that since DNA has to unwind, so the region which unwinds is mostly rich in AT sequences. Why it is rich in 80 sequences? Can you think of why it is rich in 80 sequences? Why not GC sequences? Because 80 has two hydrogen bonds, whereas GC has three hydrogen bonds. So what does it mean? It means that it is easier to break an 80 bond than a GC bond. That is why the transcription bubble essentially have 80 rich sequences so that the DNA can be unbound easily and transcription can take place. Okay. Now, some of the general features of RNA synthesis is that this RNA synthesis is somewhat similar to DNA synthesis, but there are some differences. That means the precursor molecules are ribonucleoside triphosphate and not deoxyribonucleoside triphosphates because we are synthesizing RNA here. Only one strand of DNA is used as a template. That means when we are talking about DNA synthesis, as I already mentioned, both of the strands are involved in the synthesis of DNA. Both of the strand act as a template, but here in this case, only one strand of DNA is acting as a template. Then RNA chains can be initiated de novo. That means primer is not required here. So unlike in DNA, so when you are doing DNA synthesis, your primer is needed, but in the case of RNA, primer is not needed. Then the RNA molecule will be complementary to the DNA template and identical to the DNA non-template. Okay, this is also important to understand. Then RNA synthesis is catalyzed by RNA polymerase and proceeds in 5' to 3' direction. So DNA synthesis also proceeds in the direction of 5' to 3'. RNA synthesis also goes in the direction of 5' to 3'. Then what is the difference? The difference lies in the enzyme which is used. Here the enzyme is RNA polymerase, but in the DNA, in the case of DNA, it is DNA polymerase. Now, for the opening of transcription bubble, in the case of prokaryotes, you have an AT-rich region, whereas in the case of eukaryotes, you have an autonomously replicating sequence, which is also AT-rich only. Okay. So this completes the lecture 2. Let us come to lecture 3. So, what are general features? We already discussed that it requires a DNA dependent RNA polymerase. Why it is DNA dependent? Because for the synthesis of RNA, DNA is only used as a template and not RNA is used as a template. And it is RNA polymerase because it is polymerizing RNA. Then synthesis occurs in 5' to 3' direction, so it is a unidirectional. Okay, it's not bidirectional, whereas in the case of replication, it can be bidirectional also. But here it is unidirectional synthesis. Then unwinding of the DNA occurs, not the complete denaturation of the DNA here is not observed. Now, when we talk about RNA transcription, the three major steps that are there in RNA transcription is initiation, elongation, and termination. Okay. These are the three major steps which occurs during RNA synthesis: initiation, elongation, and termination. So, what is the meaning of initiation? To initiate or the formation of the first phosphodiester. Elongation means that the movement of the transcription bubble. This transcription bubble moves, and that is when the transcription bubble moves, the RNA which has been synthesized, first phosphodiester bond which has been created, and that eventually it moves. And then termination. Termination means the termination of the RNA. It means when the RNA is completely synthesized, then the process is terminated or stopped there. So these are the three major steps which are required in RNA transcription: initiation, elongation, and termination. So now when we talk about RNA polymerase, so RNA polymerase is different for E. coli and in the case of eukaryotes. E. coli means prokaryotes. So prokaryotic and eukaryotic RNA polymerases are different. Prokaryotic RNA polymerase is somewhat simpler in its structure, whereas eukaryotic RNA polymerase is complex in structure. Okay, so this is one thing. But here, when I am uh, uh, I am talking about the prokaryotic. Here, we are discussing the prokaryotic RNA polymerases. So, prokaryotic RNA polymerase has a tetrameric. Tetrameric means 4, 2 alpha, beta, beta dash, alpha, 2, beta, beta dash, and this is known as core enzyme. So core enzyme has 2 alpha subunits, one beta subunit, and other beta dash subunit. Alpha 2 means 2 alpha subunits are there. So this is your tetrameric food. And holoenzyme requires additional sigma factor. So now this is a core enzyme. This core enzyme cannot by itself start the transcription process. So for the initiation of transcription process, along with this core enzyme, you need the extra sigma factor. Okay. And when the sigma factor comes and binds with this core enzyme, it becomes complete holoenzyme. Okay. So core enzyme plus it is known as holoenzyme. Okay. So this is important. The sigma factor is specific to genes and specific to cells also. Means that for certain specialized synthesis, certain specialized sigma factors are needed. Okay. Then alpha subunit. What is the function of alpha subunit? Alpha subunit is required for the assembly of the tetrameric cone. That means this tetrameric core can be assembled with the help of alpha subunit. Beta subunit, it is required for the ribonucleoside triphosphate binding site. Is required for the ribonucleoside triphosphate binding site. Beta dash is a DNA template binding region, and sigma factor is required for the initiation of transcription. So these are some of the important points which need to be remembered when we talk about the E. coli RNA polymerase. Now let us come to the first part, which is your transcription initiation. So what happens during the transcription initiation? So during transcription initiation, what happens? This RNA polymerase holoenzyme, it comes and it binds to the promoter, and then this, see, this is your DNA template and it has promoter. So first your RNA polymerase has to come and it has to come and bind to the promoter region. Now how it will recognize where the promoter region is present over here? That is one question. Okay. That it needs to know that where the promoter region is present. So for that, it has the sigma factor, the one which is making it a holoenzyme. So basically, sigma factor, it recognizes the promoter region. Okay. Remember that sigma factor recognizes the promoter region. So unwinding of the DNA strand takes place to provide single strand template. Then what happens? Binding of RNA polymerase holoenzyme to the DNA template. Then This is a closed binary complex. Binary means two things are there. What are the two things? First is your DNA template, and second one is your RNA polymerase enzyme. So this RNA polymerase enzyme and closed DNA template means it still has not unwounded itself. This is known as a closed binary complex. Okay. Now what happens? That formation of the first phosphodiester. Now, then what happens? DNA melting. That means uh, locally the DNA gets unwound and it forms a transcription bubble. And when the transcription bubble is formed, then it is known as an open binary complex. Okay. Up till here, it is binary only because there are only two things, RNA polymerase and DNA. But this is closed binary complex and this is open binary complex. Then what happens? Formation of first phosphodiester take bond take place, and first phosphodiester bond take place, and one first phosphodiester bond is formed, small mRNA is synthesized. So now it is a ternary complex. Why it is ternary? Because now there are three things. You have your enzyme, the RNA polymerase enzyme, you have your DNA template, and very small RNA is also there. So this is not ternary complex. So here abortive initiation can happen, or if it is not, then sigma factor is released and then the elongation is continued. For this elongation, this transcription bubble keeps on moving and hence the elongation happens. So now the question comes that why sigma factor is released after the ternary complex is formed? Why does sigma factor does not stay over there? What can be the reason behind it? Why sigma factor leaves? What do you think is the reason that why sigma factor leaves? So this sigma factor has to leave because maintaining sigma factor in the RNA polymerase and keeping it as a holo enzyme requires a lot of energy. So the only function of sigma factor was to recognize the promoter and initiate the transcription process. Now once that is completed, there is no as such function of the sigma factor. So keeping sigma factor then is energetically very expensive. Why to keep an energetically expensive component when it is not needed? So that is why the sigma factor is released after the ternary form complex is formed. So in elongation, you have the enzyme, but that is a core enzyme. That is not a holo enzyme. Why? Because sigma factor is released then. Sigma factor is not needed. So transcription initiation is essentially up till the formation of this phosphodiester bond. Once this phosphodiester bond is formed, your transcription initiation is also completed and your next process, which is transcription elongation, that starts. So now, before going on, we should know what is an E. coli promoter. So promoter is basically the place where the enzyme comes, sits and binds to it. So for the enzyme to recognize promoter, we have the sigma factor. Sigma factor comes and bind to the promoter. Now, there are two important consensus sequences which are present in promoter. The first one is the -10 consensus sequence, and the other one is the -35. Okay, and these consensus sequences are highly conserved. That means that they do not change. They haven't changed. They are the conserved regions. Okay, so now we come to transcription elongation. So when we talk about transcription elongation, what is transcription elongation now? Mm, just a minute. It is the movement of the transcription bubble. That means the transcription bubble that has opened. Now it will sequentially walks over the DNA template, and locally wherever it is, it gets acted upon, wherever the DNA gets melted, that portion of the DNA is transcribed to R. So after the formation of the first phosphodiester bond, the sigma factor is released. Then there is and unwinding continues, and it looks like that the transcription bubble is walking over the DNA, and RNA chain continues to grow, and RNA polymerase is bounded to both DNA template and RNA. So this is what transcription elongation tells. And just I'll try to close it. See, this is just a screenshot of the one of the image that is already present in your lecture. So if you're unable to see this clearly, because I can see that this image is a little hazy, then you can always go to the lecture and see and find out about it. But the concept here is that sigma factor, which we are talking about since so long, that sigma factor is important for the recognition of the promoter. This sigma factor is not there's not one general sigma factor which is applicable at every places. No, there are specific sigma factors which have specific functions. Okay, sigma 70, when we talk about, this is the general sigma factor that we talk about, sigma 70, because this is involved in housekeeping genes. The sigma 70 sigma factor is involved in housekeeping genes. Along with this, there are other sigma factors which are involved in nitrogen metabolism, heat shock, plagiarized synthesis, chemotaxis. So this is elongation. Then we come to transcription termination. Now, transcription termination can take place by two methods. The first one is the row dependent method, and the second one is the row independent method. Row dependent method means which requires the protein row, and row independent means which does not require the row protein. Okay. So row protein, when row protein is used for the termination of transcription, that is known as a row dependent termination. When row protein is not used for the termination of transcription, then it is known as a row independent termination. Okay. Just a minute. So row dependent requires the row factor and a row utilization site, and this row utilization site in short is known as the RUT site. And when we're talking about row dependent termination, then it is an intrinsic termination, and okay, I think this should be row independent. Okay, and this is row dependent, and just remove this line. Okay, I'll explain you. See, there can be row dependent or row independent. So row dependent means it requires the row protein and it requires a RUT sequence. Here it is not needed. It is an extrinsic termination, and because some extrinsic factor, which is your row factor, is required over here. Here it happens by itself. So it is intrinsic termination. Okay. Then, since it is an intrinsic termination, what happens that there is inverted repeat sequence which is present at the last, which forms a GC hairpin loop and creates a trap. Okay. So what happens over here is in row independent that some hairpin loop formation occurs in the end of the transcription, which kind of helps in the termination of the transcription process. So these are the two methods or the two ways essentially by which transcription can be terminated or stopped. One is row dependent, the other one is row independent. Then we come to the last, it's the second last lecture, I think, yes. Then we're talking about the different kinds of RNA modifications that happens during termination and during transcription, and a little bit more about the termination process. So when we talk about E. coli, E. coli has a special feature. What is that special feature? Let me just. Hmm. This image is not very good, but just to make you explain. So what happens that in the case of prokaryotes, transcription and translation are coupled. Okay, I am saying that in the case of prokaryotes, transcription and translation are coupled. That means that they happen or they occur simultaneously. So you see, this is the RNA which has been, this is your DNA template. Okay. This is the transcription bubble, and this is the RNA polymerase which is sitting on the transcription bubble. And this is the RNA which has been formed. Now, this RNA, this transcription bubble is still walking, it's still moving over the DNA. The transcription is not completed yet. Okay. You can see that the transcription is not completed yet, but still ribosome has attached to this RNA and started the synthesis of protein. So here translation is also occurring and here transcription is also occurring. Can this be possible in the case of eukaryotes? Think, is this possible in the case of eukaryotes? If yes, then why? If no, then also why? What can be the reason? Think. So, in the case of eukaryotes, we have a nucleus. Whereas in the case of prokaryotes, we do not have the nucleus. That means in the same cytoplasm, transcription and translation both will happen because we don't have a well-defined nucleus and separate compartmentalization. So there are no separate compartmentalization in the case of transcription prokaryotes. But in the case of eukaryotes, transcription happens in the nucleus, whereas translation happens in the cytoplasm. So then that means that the transcription has to be completed first. First, your RNA has to be formed, then only it can come out of the nucleus into the cytoplasm, and then only your translation can start. And because you have to have some post transcriptional modifications also. So It cannot happen coupled because the place where transcription happens is not the place where translation also happens in the case of eukaryotes. Translation happens in the cytoplasm, whereas transcription happens in the uh, nucleus. Whereas in the case of prokaryotes, since there is no nucleus, everything is in the cytoplasm, so it is easier. Okay, so that is why. So I'm just so what is written over here? Transcription and translation occurs simultaneously as there is no dedicated nucleus. That's what I'm talking about. That since there is no dedicated nucleus, so therefore it can happen simultaneously. In fact, it occurs simultaneously in the case of prokaryotes. One RNA is bound by many ribosomes simultaneously. You can see here, this is one RNA, and one RNA is attached to so many ribosomes. So one one RNA is bound by many ribosomes. Then it enhances the rate of production of proteins. So what happens? That because this is bounded to RNA which is still getting transcribed, so it enhances the rate of production. Then reduces the degradation chances of this is very important. Why? Because see, this cytoplasm has a very reducing environment. Okay, and in this reducing environment, only transcription is getting synthesized. No PTM, that is post transcription modifications, are also happening. So it reduce. So this RNA is very much prone to get reduced or to get degraded. So this RNA should not get degraded even before the formation of this proteins. So when this transcription and translation are coupled, it is reducing the chances of degradation of RNA. Okay, so this is very important. Fine. So these three processes are actually your PTM. What is the meaning of PTM? Post transcriptional modifications. This is post transcriptional modifications, and these are capping, poly A tail, and splicing. Okay, so capping में what happens? Your 7-methyl guanosine cap, it gets attached to the 5 prime. Okay, the 5 prime end में your 7-methyl guanosine caps gets attached, and it provides resistance to the 5 prime exonucleases. So this capping is important because it makes your newly synthesized RNA resistant to the 5 prime exonuclease activity. So this capping is very much important there. Just a minute. Then enzyme which is required for the addition of the 7-methyl guanosine cap is guanylyl transferase. So this guanylyl transferase enzyme is required for this 5 prime capping. Then poly A tail. So what happens? This is your RNA. This is your 5 prime end, and this is your 3 prime end. This is a newly synthesized RNA, which is not a mature RNA. It is just a it's a naive RNA. So here at the 5 prime end, your 7-methyl guanosine caps gets attached, and at the 3 prime end, your poly A tail gets attached. So this is your 5 prime capping, and this is your poly A tail. And then there is another event which happens, which is your splicing, which is the removal of introns. Okay, splicing is the removal of introns. So what happens is, see here, this is your mRNA. It has introns, introns, introns. Okay, these introns are removed in the final mRNA product. So in the final mRNA product, you only have exons, 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 and exons. This is your final mRNA. This removal of introns is known as splicing. So these introns are spliced out. Now comes the question that why these introns needs to be removed. These introns needs to be removed because your RNA only has the coding portion of the DNA. RNA only has the coding portion of the DNA. What does it mean? That only that portion is present which is responsible for the synthesis of proteins. These intronic regions are not responsible for the synthesis of proteins. These regions are only responsible for increasing the efficiency of transcription. It can, or the efficiency of the protein synthesis. Then next is, so this is splicing. So these are the three post-transcriptional modifications that happens in the case of eukaryotes. Okay. Now, this 5' capping. So this little bit detail about this 5' capping. So this 5' cap is chemically similar to the 3' end of the RNA molecule, and it provides significant resistance to the 5' exonucleases. This is important that this 5' capping, it provides resistance to the exonuclease activity. Then small nuclear RNA unique 5' cap. Okay, this is all you can study. Important is that this 5' capping, which is 7-methylguanosine, it provides resistance to the 5' exonuclease activity. Then we'll come to the last this thing lecture, which is lecture 5. After this, we'll start the questions. Okay. So this lecture 5 talks about the different polymerases which are there in your RNA transcription in the case of eukaryotes. So in the case of eukaryotes, you have 5 different types of polymerases: RNA polymerase 1, 2, 3, 4, and 5, in which RNA polymerase 2 is the one which is required for the transcription of mRNAs. So see, there are different kinds of RNAs we talked about. Just we have few slides back, we have talked about that there are different kinds of mRNA. Okay. So for the synthesis of different kinds of mRNA, different kinds of RNA polymerase are used. So for example, RNA polymerase 1 is used for the synthesis of rRNA. RNA polymerase 2 is used for the synthesis of mRNA. RNA polymerase 3 is used for the synthesis of tRNA. 4 is used for the synthesis of siRNA, and 5 is used for also some synthesis of some siRNA plus some non-coding transcripts of siRNA. So different polymerases are required for the synthesis of different types of RNA. But for the mRNA synthesis, RNA polymerase 2 is required. This is important. Okay. That for mRNA, RNA polymerase 2 is needed. Then you see that different, I'll just increase. Hmm. Here you see that different kinds of transcription factors which are required when we're talking about eukaryotic initiation. Okay, so whenever we talk about eukaryotic initiation, there are many different kinds of transcription factors which are required. See, and these transcription factors can be of two types. First is the basal transcription factors and some other transcription factors. So these are the ones which are written over here. This TFT2D, A, B, F, E, H, U. These are the basal transcription factors. And some other transcription factors, specialized transcription factors, are enhancers and silencers. Okay. So transcription factors that I'm talking about in the case of eukaryotes, there are basal transcription factors and some, and these are essentially answers and silencers. So first is your TFT2D. TFT2D, after your TFT2A comes, B comes. This is just a sequence. Important here is the helicase activity, which is uh, required in the last step. That is very much important. Okay. Now we come to the phosphorylation of the C-terminal domain. As I was telling you that this helicase activity is very much required, which is present at the last, and this helicase activity is given with the help of transcription factor 2H, TFT2H. So this transcription factor 2H has helicase activity and it has kinase activity also. So this helicase and kinase activity helps in the phosphorylation of the C-terminal domain. And remember, when we talk in the case of eukaryotes, when we talk about eukaryotes, this phosphorylation of the C-terminal domain is required for the elongation start. It is required for the start of the elongation process. In the case of eukaryotes, this is needed. And this kinase activity helps in the phosphorylation of the C-terminal domain. Okay, this is about it. So now we come to the questions. So for the questions, as I always tell, that I'll give the question and then I'll give you some time, maybe 2 to 3 minutes, to think about the correct answer, and then I'll switch the screen and I will tell the correct answer and I will explain the concept behind it also. So the only reason that we deal with the theory part is so that you get a little uh, you get it recaptured of whatever we have already dealt or you have dealt in the past week, and it will help you to understand these questions better. We can also do in this way that we'll cover week one's theory and then week one's question, then week two theory and week two question. In that way also we can do, so that it doesn't becomes boring. We have some questions also in mind. So I'll try to do that from the next class, that like if week one has like week four, this is week three, huh? If week four has say 4 lectures, then we'll do first lecture and then the questions which are related to first lecture, then second lecture, questions related to second lecture, then third lecture, questions related to third lecture, and so on. So that there is a balance between your theory and your questions as well. So, but today in this class, there were 5 lectures, and these 5 lectures are very important actually because it covers the basic concepts and some specialized concepts of transcription. Transcription is an important topic whenever we are studying molecular biology, because a lot of questions comes, and not only in questions per se. If you want to understand the basic biology and its concepts, then again transcription becomes a very important topic for it. Okay, so now we'll do some questions related to it. Okay, so I'll give you 2 to 3 minutes to answer the question. Okay. The first question is, sigma factor is not important for A, RNA transcription termination, B, recognition of DNA regulatory region, C, RNA polymerase assembly, or D, RNA transcription initiation. I'm giving you 3 minutes. Then I'll switch the screen. Okay, so I'm switching the screen now. The correct answer is RNA transcription termination. Okay, so the question was sigma factor is not required or not important for RNA transcription termination, recognition of DNA regulatory region. Just a minute. Hmm. RNA polymerase assembly or RNA transcription initiation. So it is required for the RNA transcription initiation. Why? Because sigma factor will go and bind to the promoter region and recognizes the promoter, and then only transcription can be initiated. So this is the correct answer. RNA polymerase assembly, it is required because core enzyme is converted into holoenzyme, and then only this transcription initiation can start. So this is also correct. Recognition of DNA regulatory region, yes, because it recognizes the promoter region. So DNA regulatory region recognition is also there, and RNA transcription termination. Is it required for RNA transcription termination? No, it is in fact not present up till the termination step, because it is only, it gets released after the transcription initiation only. We have already discussed that because sigma factor, to maintain sigma factor, it is an energetically expensive process, and since it is an energetically expensive process, RNA sigma factor is released when the transcription is initiated. So the correct answer for this is this one. Fine. Next question is, row dependent transcription termination require unique DNA secondary structure, unique RNA secondary structure, unique poly A signal, or row binding site. What is the correct? Think. Hmm. Think what can be the answer. One more minute. Okay, now I'm switching the screen. So the question is, row dependent transcription termination requires unique DNA secondary structure, unique RNA secondary structure, unique poly A signal, or row binding. So row dependent means what? It requires the row protein. Now the row protein has to go and bind somewhere. And what is the place where row protein go and bind some place? It is the row utilization site, which is the RUT site. So this is this. The answer would be row binding site. Okay, this is the correct answer. Okay, next question is, which of the following is gene specific? Alpha factor, beta factor, sigma factor, or delta factor? Okay, these are your options. That means that these are the factors of which, these are the factors of your polymerase, RNA polymerase RNA polymerase enzyme complex. These are the factors which are present in RNA polymerase. So, which of the factor is gene specific here? This you have to type. I'm giving you 3 minutes and then I'll switch the screen. 2 minutes more. 1 more minute. Okay, now I'm switching the screen. The question is, which of the following is gene specific? So, which of the following would be gene specific? The correct answer would be sigma factor. See, this is the core complex. Alpha 2, beta, and beta dash, it makes the core complex. And when sigma factor also comes, it makes the holoenzyme. Okay. Now, this holoenzyme, this sigma factor, we have already talked previously also that different for different functions, different sigma factors is responsible. Whereas sigma factor can differ, but this core complex remains the same in all the cases. So, which would be gene specific? The sigma factor. Next question is, coupled transcription and translation is for increasing the rate of protein synthesis, facilitating transcription termination, transcription initiation, or to create RNA secondary structure formation. So, coupled transcription and translation. What is coupled transcription and translation? When transcription and translation both are occurring simultaneously. And where it happens? It happens in the case of prokaryotes. So, I will explain that later, but okay. So, I'll give you 3 minutes. Try to answer this question. Think what can be the answer of this question. 1 more minute left. Okay. Okay, now I'm switching the screen. Hmm. The question is coupled transcription translation is for increasing the rate of protein synthesis or facilitating transcription termination, transcription initiation, or to create RNA secondary structure formation. So, what is the function of coupled transcription translation? It increases the rate of protein synthesis. Okay. How it increases the rate of protein synthesis? Because simultaneously as transcription is happening, translation is also occurring. So what happens? How does it facilitates? So because in the case of prokaryotes, transcription and translation, there is no nucleus. So everything is in the cytoplasm, and cytoplasm has a reducing environment also. So there are chances that once we wait for the whole mRNA to get synthesized and then start the translation process, the RNA might start getting degraded. So why to wait? We can, whatever portion of the mRNA has been synthesized, let's start making proteins from that portion itself. And from the same mRNA, so many proteins are getting synthesized simultaneously while the mRNA is still being synthesized. So this will increase the rate of protein synthesis. So coupled transcription translation is for increasing the rate of protein synthesis. The next question is mRNA capping ensures protection, translation, splicing, all of the above. mRNA capping. Think 3 minutes for you to think. 2 more minutes left. One more minute left. Okay. Shall I switch the screen? I'm switching the screen. Fine. The question is mRNA capping ensure protection, translation, splicing, or all of the above? And the correct answer is all of the above. How? Because your this is your mRNA and this is the 5 prime end. So when it is protected at the 5 prime end, anything which is unwanted, any unwanted event will not happen, whether it is the cutting of the mRNA with the help of exonucleases or any of the unwanted event. So therefore the answer would be all of the above. Okay, next. Next question is, CTD phosphorylation is important for A, transcription termination, B, transcription initiation, C, transcription acceleration, or D, transcription elongation. What is CTD? What is the full form of CTD? Carboxyl terminal domain. CTD means carboxyl terminal domain. CTD phosphorylation occurs where? Whether it occurs in prokaryotes or eukaryotes, CTD phosphorylation occurs in the case of eukaryotes. So I have given you two hints. Now you think what can be the answer of this question. Carboxyl terminal domain phosphorylation is important for what? Two more minutes left. Okay. So I'll switch the screen now. So CTT phosphorylation is important for the transcription elongation. So we know that once the transcription initiation is completed in the case of eukaryotes, then for the elongation, the carboxyl terminal domain is phosphorylated with the help of which transcription factor? The transcription factor which is needed is TF2H. It has the helicase and kinase function both. This kinase function, this helps in the phosphorylation of the carboxyl terminal domain. Okay, so this kinase, which is TFT2H, have kinase activity also, and this kinase activity, it helps in the phosphorylation of the carboxyl terminal domain. Is it fine? Now we'll come to the next question. Gene expressions are not regulated. True or false? What do you think? Whether gene expressions are regulated or not? Think what can be the answer. Hmm. Gene expressions are not regulated. You have 1 more minute left. Think. Class is going on. Class. Hmm. अभी है class. It's going on. Oh, you good? Oh yeah. Since 1 hour, half an hour is break. No, it's left half an hour is left. It's yeah. वो लोग ही सो जाएंगे ना मेरे जिंदगी very difficult है. Okay. But it's good that you finish in one go. वो ठीक है but it's very tiring, no? Okay, so the correct answer is false. So the question here says that whether gene expression is regulated or not. So we know that expression of gene is regulated. How gene expression is regulated? Because the expression of gene happens in cell and development stage specific manner. Cell and developmental stage specific manner. What is the meaning of cell and developmental state specific manners? So we should know that all the genes are present in all the cells. Okay. All the genes are present in all the cells, but not all the genes are expressed at the very same time. So how it happens that all the genes are not expressed at the very same time? Because the expression of gene is regulated. That means a gene which is responsible for eye color will not be expressed in your liver. A gene which is responsible for retina will not be expressed in your ear because the expression is regulated. The expression is cell specific and also developmental stage specific. Certain genes are required at certain developmental stage and they are only expressed in those developmental stages, not in other developmental stages. So the expression of gene is regulated, yes. And what molecularly, on the molecular level, how the expression of genes are regulated? With the help of certain uh, regions or certain factors, for example enhancers, promoters, silencers. These helps in the um, regulation of the expression of genes, and then there are some other methylation, acetylation, many things are there, but on the core the expression of gene is regulated. Then the next question is, TSP is the first base of DNA, true or false? Think. TSP is the first base of a DNA. One more minute left. Okay, now I'm switching the screen. So the correct answer is false, because the first base of DNA can be anything. It can be A, T, G, C, anything. It need not to be TST. So this is false. Okay, next question is, adenosine nucleotide forms the mRNA 5' cap. True or false? Adenosine nucleotide forms the 5' cap. So what is 5' capping? 5' capping is a post-transcriptional modification that occurs at the 5' end of the pre-mRNA which is being formed. And this 5' capping prevents the mRNA from the action of various exonuclease, and it also helps in the unwanted degradation of the of the mRNA molecule. This is 5' capping. It is a post-transcriptional modification. So now, what actually gets added to the 5' end is the question. Is it adenosine nucleotide? True or false? What gets added to the 5' end of the mRNA during 5' capping? Okay. So adenosine nucleotide forms the 5' cap. What gets added? Think. Is it true or false? If false, then what gets added? Okay. So I'm switching the screen now. The answer is false, because what gets added at the 5' end? At the 5' end, your 7-methyl guanosine gets added at the 5' end. Okay. So this is false. It is not adenosine. It is 7-methyl guanosine which gets added. Then RNA do not undergo maturation. Is it true or false? Does RNA undergo maturation? Think. 3 minutes and then I'll switch the screen. So shall I switch the screen? Okay, I'm switching the screen now. Okay. So the correct answer is false. That means RNA do undergo maturation. So whenever after your replication, your RNA is formed, that is not a mature mRNA. Okay. That mRNA undergoes some kinds of maturation processes to become a complete mature RNA which comes out of the nucleus. So what are the events that occurs that makes it a complete mature mRNA? So those are known as post-transcription modifications. And these post-transcription modifications are 5' capping, 3' poly A tail, and splicing. So these are the three majorly post-transcription modifications. It's not that only these are the post-transcription modifications. There are several other post-transcription modifications that occurs in the RNA, but these are essentially the major post-transcription modifications that happens in the RNA. I don't know why it gets every time. Mm, nah. So these are the three major post-transcription modifications that happens. Okay. So if the question comes that RNA do not undergo a maturation, it is false because RNA do undergo the maturation process. So now with this, we have completed the questions that are covered in the week 3. So week 3 essentially dealt with the transcription process. So in the transcription process, it started with what's the central dogma and what are the enzymes which are needed in the central dogma. And then we started with the types of RNA molecules. There are different kinds of RNA molecules and different kinds of RNA polymerases are required for the different RNA molecules to be synthesized in the case of eukaryotes. Then we talked about how the RNA transcription happens. So RNA transcription essentially involves 3 steps. First is initiation, then it is elongation, and then it is termination. So, and this RNA transcription also involves the synthesis of a transcription bubble. This transcription bubble is nothing but the DNA which is temporarily melted. Your DNA RNA polymerase is there, and your synthesized RNA is there. It forms your transcription bubble. So then we come to the RNA polymerase enzyme. RNA polymerase enzyme is a core enzyme which has 2 alpha subunits beta and beta dash. And when sigma factor also comes and joins in it, it becomes a holoenzyme. Then we come to the initiation of transcription, how transcription is initiated. So first your RNA polymerase comes and it binds to the DNA. It is a closed binary complex. Why it is binary? Because RNA polymerase and DNA only 2 things are there. Then this closed binary complex is converted into an open binary complex when DNA gets melted locally. And after that, your first phosphodiester bond forms and it becomes a ternary complex. After the formation of first phosphodiester bond, your sigma factor is released and then it goes for the elongation process. In the elongation process, what happens that a transcription bubble is there. That transcription bubble, this transcription bubble keeps on moving and hence the elongation takes place. And then for the termination, we have row dependent and row independent termination. Row dependent means it requires a row protein and a sequence, that is red sequence, and it is an extrinsic way of the termination of transcription. Whereas row independent, it is an intrinsic way of the termination of transcription. It does not require the row protein, it does not require a red sequence, but there has to be some hairpin or stem loop structure which is rich in AT sequence. Again, the logic for the AT sequence remains the same. If there is an AT sequence, because AT has 2 hydrogen bonds, it is easier to break a AT sequence than to break a GC, because GC has 3 hydrogen bonds. So this kind of a hairpin or hair loop structure, what it does, this hair loop structure kind of helps in dragging and it helps in the termination of the transcription process. So this is your row independent sequences. Then we talked about coupled transcription and translation in the case of E. coli. That means in the case of E. coli, in the case of prokaryotes, basically, the transcription and translation processes are coupled. So they occur identically because there is no dedicated nucleus. So once the RNA is bounded, you can see this mRNA is still getting formed. And this mRNA is getting formed and the RNA is still bound with so many ribosomes here. Simultaneously, RNA is bounded by many ribosomes. It enhances the rate of protein production and reduces the degradation chances of the RNA also. This is your coupled transcription and translation, which occurs in the case of E. coli. And then the three different kinds of post-transcription modifications, not in detail we have dealt, we will deal or learn about these uh, modifications in detail in the subsequent classes also. These are 5' capping, poly-A tailing, and splicing. So in the 5' capping, your 7-methyl guanosine cap is added, which is chemically similar to the 3' end of the RNA molecule. It provides resistance to the 5' exonuclease activity, and the enzyme which is required for this 5' capping is guanylyl transferase. Poly-A tailing, your 20 to 200 nucleotide poly-A tail is added at the 3' transcript, and the enzyme is poly-A polymerase. It is important. The enzyme which is needed for poly-A tailing is your poly-A polymerase. Then splicing mein, your introns, which are the non-coding parts, are removed. And then different kinds of polymerases. In eukaryotes, we have different kinds of polymerases for the synthesis of different types of RNAs. But RNA polymerase 2 is needed for synthesis of the mRNA. This is important. mRNA synthesis mein RNA polymerase 2 is needed. Then for rRNA, RNA polymerase 1 is there. For tRNA, 5S RNA and other small nuclear RNA, your RNA polymerase 3 is there, and 4 and 5 are basically for your plants. Then this is important. In the case of eukaryotes, the carboxyl terminal domain has to be phosphorylated. After the phosphorylation of the carboxyl terminal domain, only your elongation process will start in the case of eukaryotes. So you see here that these are the different kinds of transcription factors. Yes, in the case of eukaryotes, you need several transcription factors, and these transcription factors are classified as general transcription factors or basal transcription factors and specialized transcription factors. Specialized transcription factors can be your enhancers and silencers, and basal or general transcription factors are these ones. TFT2H, A, B, and all these things. And these, amongst these, TFT2H is important because it has helicase activity as well as kinase activity. This activity is required for the phosphorylation of the carboxyl terminal domain. And after the phosphorylation of the carboxyl terminal domain only, it will undergo the elongation process. Okay. So is it clear to everybody? I hope that this lecture is clear. The recordings will be available to you. You can go through the recordings and see if you have any query or any problems. You can let me know, and we can find out the answers of that. And practice as many questions as you can. That is only going to help you. So, okay. Then we'll try to solve even more questions. If you have any other queries, you can come to me and we can uh, just a minute. We can come and we can try to solve them also. Okay. So then we'll stop the class here. So next week we'll meet on Tuesday from 7 to 9, the same timings. Okay, fine. Bye everybody.